But we can look at that later in lab, what is it, 4, and look at microscopes. All right, so what we were talking about the other day, any questions, anybody, before we go on? Any problems, anything we need to get out of the way first before we get into material? We're all cool? Okay. Let me update my role here just to make sure. So I got everybody credit for. All right. So last time we were talking about different types of organic molecules, and we call them classes of organic molecules or groups of organic molecules. And the first one we talked about was carbohydrates. And on each one, what I tried to do initially is we talked about, what their purpose was, what do we use carbohydrates for? And then I'll mention some of the monomers, maybe not necessarily all of the monomers, but just kind of a representative because, if you remember from our discussion, the big thing about carbon organic chemistry is diversity, right? So there's many monomers. And then again, some of the more common polymers, perhaps not all of them, but at least the large groups of them. So the next group of organic molecule that I want to discuss are the lipids. So let's do that first thing first. What do organisms use lipids for? And if you're not sure about the term lipid, you can see in the descriptions, these are fats and waxes, oils, that kind of stuff. Hydrophobic things, by the way, right? Hydrophobic things. So give me some examples of how lipids are used by biological critters. Or biological, I always say critters because I'm so zoological, because that was my degree in zoology. Yeah, you can find lipids in the blood, but give me an idea of the purpose. Give me an idea of the purpose. Now, the lipids in the blood are probably lipids that are being transported around from things that people, organisms eat, right? So give me and think about ways in which animals usually use lipids. What do they use lipids for? I mean, what are fats used for? Yeah. Right. Remember the other day yesterday I gave you the analogy of energy in your bank account, and I said the money in your pocket is like the sugar in your bloodstream, or the energy molecules in your bloodstream, or the food, right? The stuff in your liver would be like in your checking account, and the stuff in your fat, I said, would be like long term. That would be your savings account, right? So fats are long-term energy storage. Back in the olden days, right, and I'm talking about when we were running around as cave folks. Meals weren't, well, they didn't necessarily have 3 squares a day, right? Didn't have 3 meals a day. They might get a meal today, not such a good meal, maybe a little bit for dinner, and then tomorrow, oh boy, they took down a mammoth. There's a big meal we're going to eat for quite a while, and then holy cow, where'd all the mammoths go? Not much to eat. We're eating lizards and rats, right? So fat is a way for organisms to store for those leaner times. Something much more applicable to wild critters than us, right? Because we typically don't go without eating for days on end unless you're intentionally doing that sort of stuff. Okay, so yeah, long-term energy storage. But that's remember in the sugars and the carbohydrates, I said that typically it's energy, but then other types like cellulose, which is a carbohydrate, cellulose had a very important function. Not so much energy. Now, you can get energy from cellulose if you're the cow and have the right bacteria that live with you. But remember, cellulose was also what's used to make cell walls of plant cells. Cell walls are very firm. It allows plants to stand up, right, because they don't have a skeleton. So can you think of any ways in which lipids are used by critters out there that aren't energy related, but used for another purpose? Now, living in Florida, I can give you a little bit of a hint. The part that says waxes, if you're familiar with red mangrove plants. Yeah, warmth. Okay, give me an example of an organism that uses lipids for warmth. In those critters, we might call it blubber. There you go. Whales and dolphins and manatees, they all have a lipid layer, and indeed that's exactly what it is. It's a storage of energy, but it also provides an insulation for them. Now, when I worked at SeaWorld, one of the ways that we could tell that an animal was emaciated is by looking at the blubber layer. And the thinner the blubber layer, the more emaciated the animal was. And the additional problem with that is being a warm-blooded critter living in water, those critters have less warmth, less insulation. If the water turns cold in the winter, they're the first ones that are going to feel the effect of the cold weather, right? So yeah, insulation. How about the red mangroves? The ones with the big prop roots. The big. You see them on the Indian and Banana Rivers down in the Keys. You know, it's those. If you ever get a chance to snorkel around those, have the cojones to go up in underneath those prop roots. You'll see incredible things. Fish nurseries. A lot of fish will lay their eggs near the near those prop roots, and they drift up underneath so that the little ones can grow up before the predators come get them. So if you can kind of make your way underneath there, and it's a little bit dark and spooky, but you might come across one of these places where all these one species of fish, little tiny ones, are all hanging out. It's like so cool. Snakes where? In red mangroves? Yeah. There are. Well, I would say they usually get out of your way, but moccasins aren't so accommodating, but yeah. All right. Anyway, the leaves of red mangroves, the next time you see one of those, put it in your hand and feel it. It's got a waxy coating to it, right? It's got like basically it's chapstick. Or plants that have that waxy coating, or in function it's chapstick, are plants that have to worry about losing water, about dehydrating, which is what you use chapstick for, right? Make your lips all soft and kissable and all that kind of stuff. But it basically is wax that retains water, keeps your lips moist. Red mangroves grow in water that is not salty, but not fresh. Is what we call brackish. It's in between. So they have a concern about dehydrating in salt water. So that's what their leaves are. Well, if we're going to have to worry about the salt water, dehydrating us, at least the leaves can trap some water in, right? Okay, and we'll talk about some of those other polymers as we get into them. But I do want you to understand that in this diversity of carbon molecules out there, that there are primary functions, if you will, and there are dozens and millions of secondary functions. So that's, again, that whole diversity thing, right? Okay, let's take a look at a triglyceride. All right, triglycerides, I guess I shouldn't have a comma there. Triglyceride is maybe a fancier name for a fat, okay? Triglycerides are fats. But what I really want you to notice about the word triglyceride is the tri. What's that mean, tri? Excellent. Okay, take a look at this next thing. Let's put this together. Here's the tri part of the triglyceride. Okay, first of all, let me unconfuse you. Down below where it says fat molecule, see that triacyl glycerol? Same thing as a triglyceride. Same word, same thing, just a different name. All right, just a different name. Okay, let's take a look at the top. Remember what I said about all these organic molecules that we're going to go through. Now, if you let me bounce back a couple slides so I can spark your memory. Remember, I said that all organic molecules have monomers that make up polymers, and we talked about how that chemically happens. Remember this? Right? We talked about how these things are put together and how they're torn apart. And hopefully between yesterday and today, you've committed it into your head which one is which, right? And I gave you a way to remember. Hydrolysis is to split, water splitting. So that's how they're broken up, right? And I showed you how that works in the carbohydrates right there. Okay, there's dehydration synthesis. Hydrolysis would be the opposite. And now I want to show you how this works here. All right. So here's what we got. We got two monomers up at the top. We have a fatty acid or a palmitic acid. In this case, it's a palmitic acid. There are many types of fatty acids. And then we have a glycerol. Glycerol is pretty simple. Now, before we actually look at the process of dehydration synthesis, let's see how many of y'all remember from what we talked about yesterday in functional groups. Which functional group is that? If I circled that on the exam, I said, name that functional group. Even if you didn't know, you said, oh man, I'm drawing a blank. What would you put? Hydroxyl. Very close, very close. Someone always asked me a while back, says, does spelling count? Now, I'm not a real, smart ass answer is, well, yeah, it always did. I'm not a real stickler on that. When I look at spelling, I'm looking for something that's in the ballpark, all right? So Raymond, your answer hydroxide on an exam, you wouldn't have lost all the points, right? I want to give you a little partial credit, but do try to remember the right names, because a hydroxide is this is a hydroxide. Remember in acids and bases? That's an O. That's a hydroxide. Okay. Okay, so yes, those are hydroxyl groups. Now take a look at what's happening. All right, let me erase this for a second. Let's keep going. Here's a functional group right here. Oh, come on, pencil. What's that one? I didn't mean to get the second carbon, just that carbon double bonded to the oxygen. Which one is that one? Who else is in the room? Let me see. Josh, how about you? Or Wyatt? It's carbon double bonded to an oxygen, and then it's got, there you go, carboxyl. All right. So when we talk about what do functional groups do, and I said they impart function, they impart a characteristic to a molecule. In this particular case, here's an example of a very important function of functional groups, right? The two functional groups there, the carboxylic acid and the fatty acid. Remember, carboxyls can be called carboxylic acid too, fatty acid, right? They're acids, yes. And the hydroxyl that's found on the glycerol, the function they're going to perform is like the coupling on a train between two train cars. That's what it's going to do. That's going to be the linking part, okay? Now, it's going to form something that specifically is called by chemists an ester linkage. You see it down below with the red square around it. Don't worry about that too much. The characteristics of an ester linkage and why it's called that. Again, this is not a chemistry class. But notice that there are 3 ester linkages that are created, because when you make a triglyceride, you take a glyceride or a glycerol and you 3 times put a fatty acid on it. So down below you get a triglyceride, or that other word that doesn't really roll off the tongue as well. Triacylglycerol. I like triglyceride better. All right, so that's a fat. Big molecules. Fats can be large molecules. Generally have higher energy content than the other things you eat because of their size, because they are so big, okay? All right, now, modern human beings, modern Western human beings, modern human beings that aren't in the developing parts of the world, as I mentioned before, typically we don't go without meals, right? Typically we don't have to worry about eating a wildebeest today, and then maybe in 6 weeks we get a couple of rats along the way that we have to eat another wildebeest, right? So we don't worry about that sort of stuff. But fats are in our food. I mean, really, when it comes right down to it, with all the health problems in fat, in food, or with fat, they could seriously reduce. You do need some fat, by the way, but they could seriously reduce the amount of fat in food, and yet McDonald's French fries. Oh wait, they changed that, didn't they? Oh my God, but those are still awesome French fries. But what does fat do for us in food? Why do you think they put fats into foods? Sometimes they do. They actually add a fat to food. Yeah, that's one of the reasons. Sure, makes us feel satiated. You've ever been on a diet? Now, I love Oreos. Okay. Ah, there you go. I love Oreo cookies. My father was an Oreo fiend. I'm an Oreo fiend. I just love Oreos. I can't eat the chocolate as much as I used to for reasons because I'm old and entropy and all this manure. But if you're an Oreo file like me, someone who loves Oreos, oh God, yes. Holy cow. They make red velvet Oreos. See, I don't even walk by them anymore because they have all those great ones dipped in white chocolate. I just want to, and I know it'll just mess up my gut. But anyway, if you've ever been on a diet, you might have substituted your Oreos that you like for those Snackwell cookies. You ever seen that Snackwell stuff that looks like an Oreo? It's got the 2 chocolate things with this. I'll bet. But have you ever seen those? You know what I'm talking about? The Snackwell cookies. Any of those, diet cookies that they have out there, do they ever taste as good? I mean, does a Snackwell, does a Snackwell Oreo taste the same as an Oreo? Gosh, no, they don't taste anywhere near the same. Now, the Snackwells are better for you because they're low fat, and the Oreos, that's right, bootleg Oreos, man, pseudo Oreos. And the Oreos ain't worried about fat because that's what they're selling, folks. Because just like, was it Raymond that said it? Yeah, taste. Fats make things taste good. Take a look at these 2 things. On the left-hand side are saturated fats, butter, right? Everybody loves butter, right? My wife likes to cook, and she watches all these cooking shows, and I hear these chefs out there and they're going, if you're going to cook something and you're going to make something really nice, you don't skimp on the butter. Don't go with margarine or anything like that. I'm like, yeah, I get it. It does make things taste better, but the problem is it's worse for you. So saturated fats taste better, but worse for you health-wise. Unsaturated fats, canola oil, well, just the fat that you might find in those Snackwell cookies, better for you, but not quite as tasty, right? Now down below, and I put the little crossbone and the pirate thing, okay? There's this thing out there you'll read on your ingredients sometimes called trans fat. You're like, well, what does that mean? Well, here's what they've done with a trans fat. They take a good fat, unsaturated, and they turn it into a saturated fat. Now, we're going to talk in the next slide the difference between those. There is a chemical difference. You can see down below, for example, unsaturated fats are going to be more fluid, more bendy. That's what we say. Unsaturated fats at room temperature tend to be liquids. And now, if you remember your chemistry from the other day, you can tell me why that last fatty acid, whoops, oh, I just gave away, doggone it. You can tell me why that last fatty acid is bendy if you look, oh, come on, if you look right there. What is that I just circled? Yeah, it's a double bond. And what do we know about double bonds versus, say, single bonds? What do we know about the two atoms in double bonds? What can they do versus ones in single bonds cannot do? Look at the difference between those two fats and yeah, right, they're bendy. There you go, they're bendy. All right, very good. So now let me show you the difference between a saturated and unsaturated. So before I leave, again, what they do with trans fats, so that's what those are, is they take the fat that's not bad for you, unsaturated, and they hydrogenate it. So they make it saturated. You see, the term saturated and unsaturated refers to hydrogens. A fat that is saturated it contains all the hydrogens it can. And one that's unsaturated is missing some hydrogens. All right, now that's a little bit vague. Let me show you what I mean by that. Take a look right here. Oh, you know what? Sorry, folks, give me 1 second. I actually need to share something with you because in order for this to work right, I have to. Sorry, I'm actually trying to use all this stuff for an advantage, and then. Okay, no, that's not it. Right, live, share. Oh, man, you're only seeing part of it, right? Oh, geez. Thanks for bearing with me, folks. Let's see if this helps. Come on. No dice. All right. Well, you know what? I'll have to do it by hand to draw it in. That's okay. Sorry about all that. Let me get out of this. Of course, it wouldn't. I've got a long laundry list of things to tell the e-learning Collaborate folks about this. I do like this online stuff because I can throw in these videos, but man, I tell you, these programs are just. You just don't seem to handle it the way they should. All right, back to this screen right here. Let me go ahead and just old school it in. All right, now this has already come to the point at the end of the slides. So let me show you how this works. You see these double bonds that are in red? Yeah, Raymond? Anybody else having problems with the internet? Let me see how you're connected, see if it's... Oh yeah, Raymond, it says the connection's good over on this end, so hopefully it'll just be a temporary thing. Okay. All right, so you see those double bonds that are there? Imagine for just a second that those double bonds aren't there. Let me go ahead and just kind of pencil it in, because like I said, what you're seeing here is the end of my story, because with my PowerPoint, it's animated, but I can't do that here. That's why I was trying to share the application. So that right there is not a double bond right now. It's a single bond. That's a single bond. Sorry, it's supposed to be a line. That's a single bond. So now, of course, if you remember, we talked about carbon is most stable when it's sharing 8 electrons. So let's make sure that we get that for those carbons. There's got to be a hydrogen here, and up here too. Okay, and I'm sorry, it's real squiggly. It's very hard to write on a slanted screen, by the way. But you see what I got there? You see now those bonds between the carbons are single bonds, and then the hydrogens are, make up the complement. Now, this is a saturated fat. This is a saturated fat. A saturated fat is holding all the hydrogens. The one I drew in black, that's what I'm talking about, hydrogens that we're missing in the other one. But a saturated fat is full of hydrogens. Okay, now if you're looking at it going, oh my gosh, how am I supposed to know it's full of hydrogen? It's very, very simple. An unsaturated fat is missing hydrogens, okay? And an unsaturated fat, the way you can tell that it's unsaturated is look for the double bonds. If there are any double bonds in these fatty acid tails, now not this one here, not the one with the oxygen, I'm not talking about that one, but if there are any double bonds in any one of those three fatty acid tails, right, or lengths of carbon, any double bonds, it's unsaturated. If there's one double bond, mono unsaturated. If there's more than one, poly unsaturated, okay? So again, I'm sorry that was crude. It's a lot smoother with my PowerPoint presentation, but sometimes it depends on the scheduling when you get to see it, either in class if you want. I'll be more than happy to show you the animation in class if you need to see it. So remind me about that, and I will be, I'll gladly do that. But does everybody understand what I mean by saturated and unsaturated? So if you're making yourself study notes, sometimes it's best when you're looking at concepts to whittle it down to its simplest fact. And your study notes say, saturated, no double bonds, unsaturated, double bonds. And just remember that the double bonds I'm talking about are the ones between the carbons. Now, the reason why that fat in the previous picture was bendy again was because of the fact that it has double bonds, right? So those double bonds can only exist, so those double bonds can only exist if you have an unsaturated fat, okay? That's why it's bendy, just like we saw in the picture. Now, this unsaturated fat in the picture would be an example of a mono unsaturated. It's only got one double bond. Is that clear to everybody? Piece of cake, right? Yeah, they are, but don't count those as far as determining saturated or unsaturated. We're talking about double bonded carbons, and that's double bonded to an oxygen. So we're going to be looking at, only carbons. Only carbons. All right, now let's go back and review some very basic things before I move on to the next concept or the next type of fat that I want to discuss with you. Are fats hydrophilic or hydrophobic? Yeah, they're hydrophobic, right? So if you put them into water, they would not mix, correct? Because water is not hydrophobic, it's hydrophilic, correct? Everybody good with me on that? Okay, now, what if we took this, a fat, saturated or unsaturated? It so happens it is an unsaturated fat that I'm gonna introduce you to next. But let's just talk about this for a second. Let's say we take one of these fats, and instead of this fatty acid tail that's down here on the bottom, right? Let's replace that polar part of the molecule. I'm sorry. Let's replace that non-polar part of the molecule. Hydrophobic, non-polar mean the same thing. Okay, so let's take that hydrophobic or non-polar part of the molecule and replace it with something that's polar. That's hydrophilic. Now, first of all, let me ask you a question. Is that possible? Are there molecules out there, one molecule that has a hydrophobic part and a hydrophilic part? Yeah, there is, aren't there, Tim? Give me an example that we talked about in class of a molecule that has hydrophobic and hydrophilic parts on the same molecule. Laundry detergent, right? So whereas if you put a hydrophobic substance into water, it wouldn't mix. And if you put a hydrophilic substance into water, it would mix. What would one of these, remember what the word was for something that's both polar and non-polar? You guys remember that word? Right? Amphi means dual. Phile means it loves both. Phile is to love, hydrophilic, right? So if I did that to a molecule, I might be able to create something like this, right? Take a look on the left. That's exactly what I've done. I've taken one of those hydrophobic tails off. There's two there, should be three if it's a fat. I've removed one of them. Notice the double bond. There you go. That's why it's bendy. Okay, I've removed one of those hydrophobic tails and I've replaced it with this thing up here. One of your functional groups, right? Which functional group is in the yellow there? It's real easy. It tells you. Okay. And then this group of atoms with a nitrogen in there. Now, as you know, nitrogen's a lot like oxygen. So when you put nitrogen and oxygen with other atoms, especially if they're hydrogens, very often you're going to get polar parts to it. So notice that little charge that you see up there? So now what we have, all in the gray there, is have what we call a hydrophilic head and a couple of hydrophobic tails. Now, the picture in the middle is that space-filling model that I talked about the other day. You can see the phosphorus atom is there. These are the oxygens. That's the nitrogen that I talked about. And the black ones, of course, are the carbon. Now, as time goes on, what we're going to do is we're going to see these molecules over and over again. These molecules are a type of fat known as phospholipids because they have the phosphate group and then the lipid tails. That third picture, the 1C, that's the way in the future you're going to see these. So what we've done throughout those A, B, and C is just kind of made the picture simpler. But I needed you to understand the structure first. That's why I showed you these, so you could understand where the hydrophilic part came from. Okay, now, what we didn't talk about fully, there's the detergent, right? Same kind of molecule, that amphiphilic molecule. What we didn't talk about is what if you threw a bunch of phospholipids into a glass of water or a beaker of water? What would they do? Would they dissolve or would they not dissolve? Would they be attracted or would they be repelled? What do you think? If I took a bunch of those phospholipids and I dumped them into a beaker of water, what are they going to do? Hypothesize for me. If you're looking for a little hint at what I'm getting at, take a look at the picture in D. See what they call a phospholipid bilayer. Imagine the picture in D, the blue is water. Well, not so much dissolve, Tim. Not so much separate. They're going to kind of do both. Okay, that's a better word for it. As you notice in that picture there, the polar parts, the part in gray, the hydrophilic heads, they're arranged facing the water. Perfect, right? They're attracted to the water. Notice where the hydrophobic tails are. They're in that layer in between. They're repelled by the water. Now, you couldn't just have a phospholipid monolayer, because if you only had one layer of those phospholipids, those hydrophobic tails would be suspended in water. That would be problematic. So when you put phospholipids into a glass of water, into a beaker of water, what you get is a bilayer, okay? Now, we live in a three-dimensional world, so that bilayer is going to make a bilayered sphere. Like you see in the picture down below, water on the outside, water on the inside, the head's facing the water. Remember the inside of your cell that we talked about the other day briefly, the cytoplasm? There's a lot of water inside your cell. So those hydrophilic heads are going to be facing that. And the hydrophilic heads are going to be facing out where there's a water, because the world is an aqueous world and we live with a bunch of water. And they're going to form a lipid bilayer. Now, you might be thinking, why in the heck is he getting so excited about this? Well, when we get into our next chapter, chapter 6, and into chapter 7, we're going to be talking about cells. We're going to be talking about the parts inside of a cell and what cells do. And then we're also going to be talking about that layer, that border that defines a cell, or what we call the cell membrane. The cell membrane is made up of phospholipids in a bilayer. Again, if you think about the way life evolved, water was already here, okay? So if an organism was going to evolve, and one of the ways that organisms found that worked when they were evolving was all of that really cool biological dance of chemistry that we've been going through. If you contain that in a closed space, you made it more efficient. Do you remember why I said molecules come into contact each other out there in the world? I mean, how does molecule A and molecule B come together? Do they send each other an email, a text message, an Instagram? What do they do? How does molecule A, that's over here in part of a cell, and molecule B, it's over here in another part of the cell, maybe on opposite sides, how do they even get in the close range to each other to do a chemical reaction? They can't do a chemical reaction across the expanse of a cell. Well, that might cause the chemical reaction, Raymond. Yes, that might cause, like, for example, an ionic bond. But my question is, even preceding that, how do those two ions, in your example, how do they get close enough to one another to even stick? Sodium and chlorine won't attract each other from the other side of the cell. So how do those two molecules happen to get in touch with one another? Don't think too deeply about this, folks. Yeah, they're always in motion, right? Remember we said that's why heat helps. Heat increases their motion. So since they're always in motion, that means that there's a good chance, it's all random, but there's a good chance they're going to bump into one another. Now, if you confine those A's and B's into a smaller space by a phospholipid bilayer, guess what? Instead of A and B trying to find each other in a gymnasium, A and B are trying to find each other in a broom closet. A lot better chance of finding each other, right? You understand what I mean by that? So as life evolved, its efficiency increased, and one of the ways it did was to confine those incredible reactions, where we're going to get into a lot more photosynthesis and respiration, all that stuff, inside a closed space. And these phospholipid organic molecules, which are not created by a living thing, very conveniently fit the bill, right? Very conveniently fit the bill of making this package for you. So what this ball that you see is going to be a cell, right? Inside is going to be the nucleus and the chloroplasts and the mitochondria and all the stuff that we're going to talk about. Pretty cool, huh? I'm always fascinated by how these little tricks of chemistry, how life used those to engage in evolution and this whole idea of becoming efficient and just being alive. Okay, we got one more type of lipid to discuss. Any questions on that before we move on? That phospholipids. We will come back to those and talk about phospholipids quite a bit when we get into, the cells and cell structure, membrane structure. I showed you a molecule similar to this the other day when we were looking at the lions. Remember? I said that don't worry about what these rings of carbon, hopefully you recognize those as that chemistry shorthand that I've been talking about. But I said don't worry about those rings of carbon, the ones in the orange right now, because I'll talk to you about it later. Well, that time has come. So any molecule that has these 4 rings of carbon fused together like that. Now, 3 of them are 6-sided. There's a double bond right there, right? And one of them is 5-sided. Any molecule that has those rings, this one, this one, this one, and this one, regardless of the rest of this stuff, there's a functional group, there's a functional group, there's a couple of functional groups. Those are all methyls. Don't forget the methyls. They're not very exciting. They're just carbon and 3 hydrogens. But any molecule that's got those 4 rings of carbon fused together is considered a steroid. Now, there are protein steroids and there are lipid steroids. Obviously, this is a lipid steroid. You have come into contact probably several times. Well, I know you've come into contact with steroids in your life. There they are, the sex hormones. Sex hormones are steroids, aren't they? Now, look at a couple of the other ones. Prednisone over on the left-hand side. What is prednisone prescribed for? Anti what? Okay. And if you were prescribed prednisone, you know that little thing that they always stick in the prescriptions that gives you all of the details about the pharmaceutical you're taking? It's usually a little piece of paper that's been folded 1,000 times, and you open up, it's the size of a poster. They always have, or many times, every time I've seen it, they have the chemical structure If you are prescribed prednisone or know somebody or whatever, if you look at the chemical structure of prednisone, you'll now be able to tell, oh, I know what that is. It's a steroid, isn't it? See the rings? Now the rings are arranged a little bit differently, but just, artist interpretation. 1, 2, 3, 4. Androsterone is another hormone associated with being male. And look what we got here. Boom, steroids. So again, don't worry about the stuff that's attached around it, the double bonded oxygens and the functional groups. A lipid steroid is defined by those 4 rings fused together, okay? That completes our discussion about lipids. We're going to move on now to proteins. Any questions before we go on? No? All right, proteins. Oh my, life is all about proteins. Proteins, you've heard it said before, they're the building blocks of life. Lipids, we talked about energy, insulation, carbohydrates, energy, some cellulose. Proteins are the structural framework of life. Proteins, although you can use them for energy, typically not supposed to, or not a lot, but proteins are the structural framework that living organisms are built around. Your skin is made of proteins. Your body is made up of proteins, okay? The monomers for proteins, all proteins have the same monomers. Now, there's different kinds of these monomers, but they're all called the same thing. They're called amino acids. Now, I want you to look at that word for just a second because it's very important, because one of the things that I want you to know is that all amino acids, there's about 20 of them, I'll show you in just a moment, have at least two functional groups. And the name amino acid tells you which ones they are. All amino acids have an amine. Now, the amine functional group was distinct and unique. What's unique about the amine functional group? What does it contain that no other functional groups contain? So that when you're studying them, you'll remember it. Anybody? It was the only one. Remember I said phosphate is easy to remember. The sulfhydryl is easy to remember. It's the only one with sulfur. How about the amine functional group? What atom does it contain so that when you see functional groups, and you'll know it's an amine? You see what I'm drawing? I just drew an amine functional group. What atom does it contain that none of the other ones contain? There you go. Okay? All amino acids have that functional group. The other functional group they have is an acidic functional group. You've only got one, this one, the carboxylic acid or the carboxyl. All amino acids, by definition, their name, amino acid, tells you that they all have those two functional groups. They may differ in many other ways, but they all, and you're going to see in just a moment why they have those two functional groups. It's very important. All right, there are several different polymers of proteins, hundreds of thousands of them. So rather than even list any of them, I put a picture of a protein, that space-filling model, because I want you to understand something about proteins. This is going to be very important, not only in this chapter, but in subsequent chapters. You might want to write this in very bold letters in your notes. The shape of a protein is very important. Okay? Proteins can only do their proper function if shaped properly. Now, let's talk about shape for a second. See that polymer in the picture there? How would you describe that shape to somebody? What would you tell them it looked like? Which is it, square, round? How would you describe it? Doesn't have to be technical or scientific. Well, Raymond, if you saw something like that in a picture and you were telling a friend of yours, it said what it looked like, you'd say, well, it looked like a what? Looks like a fat S. Yeah, kind of, I guess. If I was walking across the floor and dropped some dough onto the floor, plop, what kind of shape would that dough make when it hit the floor? Clump, clump. There you go. A clump or a glob. Glob in another word for a clump, a glob, right? Glob. Many proteins are called globular. I don't know if I spelled that right, but whatever. Globular proteins. Okay. I want you to understand that even though to us this shape looks like a clump, right? A glob, it looks without definition. Okay. Doesn't look like a square or a rectangle or anything like that. Even though that shape doesn't look anything to us that we could describe, to Mother Nature, that particular globular shape is very important, okay? A lot of the things we're going to talk about in proteins have to do with maintaining that proper shape. That's why I want to emphasize that, okay? We'll talk about more details on the shape in just a second, but I want to do what I've done in the previous slides. I want to do a little form and function thing. I'm not expecting you to memorize all these examples here and anything like that. I really, a lot of this stuff we're going to be actually talking about in subsequent chapters. So this can be really nothing more than kind of an overview. But proteins do many things. There are enzymes that are proteins or proteins that are enzymes. All enzymes are proteins. And enzymes make organisms more efficient, able to digest food. And we have a whole chapter to talk about that. Defensive proteins like antibodies that protect you against diseases and viruses. The white part of the egg, albumin, that is ovalbumin that you see there, that is a protein, okay? And that is going to be, you know, when an organism, an egg is growing, it's going to need these proteins to grow, and that's the source of it. Mother Nature, or the mother, provides that for them. We've talked a lot about things moving across cell membranes, right? We talked about that in the context of surface area to volume ratio. And one of the ways that things can be more efficiently moved across the cell membranes is using what's called a transport protein. And we'll discuss a lot more of that in the chapter about membranes here in a couple of chapters, okay? Proteins can also be hormonal, right? I said there were lipid and they're also hormones, I'm sorry, steroids. And so hormonal proteins oftentimes are also steroids. Oh, receptor proteins. That's really cool. That's a synapse there. You see in the picture there, folks. The little microscopic gap between your nerve cells. We sometimes joke and say, oh, his synapses aren't firing. It's like somebody's not thinking. And what happens is the way the signal gets across that little gap is a protein is produced. Those molecules produced, and the receptor proteins with their specific shape, by the way, absorb those molecules, and that's how the signal gets relayed. A lot of antidepressant drugs have to do with controlling those signaling molecules in that little synapse. Your muscles are able to work because of proteins, and as I mentioned before, again, structural proteins that provide structure to the organism. So that's just kind of a quick overview of proteins. Like I said, many of these we'll go into more detail as we start to get into the specifics of their function. Okay, let me show you an amino acid. All amino acids have those two functional groups, right? An amine and a carboxylic. All amino acids have an alpha carbon, the one in red in the middle. The other side is attached to a hydrogen. All amino acids, okay? Where they differ is in that R group. Remember, there's no element R. That's like the X. Remember when I was showing you cis and trans geometric isomers the other day? There's an X, X. There's no element X. There's no element R. An R is the side group. If you wonder why they call it R, it means the rest of the molecule, the remainder of the molecule. So this is where the different amino acids, this is where they're going to differ. They're going to differ in the side group. So let me show you the amino acids. Again, I don't expect you to memorize the names of amino acids, right? I'll give you all this stuff. It's more the overall concept. Look at the colors. all of the amino acids, the part that's in purple, all of those are the same. Now, in a world with water, those that amine and that carboxylic functional group, they become charged. That's why you see in the glycine an H3N plus. See, this is the amine group, and then this is the carboxylic acid, and it's lost its hydrogen. But that's still the group that it is, okay? Those all are the same throughout all those amino acids. Now, where they differ is the part that's in the yellow, right? Glycine is very simple. It's just a hydrogen, but some of them get a little more complicated. Look at tyrosine, for example. It's got a ring in it. Notice, too, that these amino acids are clumped into groups based upon their hydrophilic or hydrophobic. The ones at the top, glycine, valine, the ones in yellow, those are all hydrophobic. The ones in green are hydrophilic for reasons because they have polar side chains. And then the ones that are acidic or basic, they're also hydrophilic because they're charged, right? If you're charged, you're hydrophilic because water is charged, and that's what attracts it to each other. Now, as we go through our discussion about the structure of a protein, right, and the shape of a protein being so important, I want you to remember that these different amino acids have different side groups, and where you put them in a chain, right, where you put them in a chain is going to determine how that protein is going to fold. The hydrophobic parts are going to want to fold away from the water. The hydrophilic parts are going to be attracted towards the water. Okay? That is all going to make that glob, that thing that doesn't look like much to you and I, but is indeed a glob. Now, let's go back and do that thing that we've done all along. Let's look at how the monomers, right, all of these, let's look at where they're connected. This is back to that purple part again. The reason why all amino acids have the amine and the carboxylic acid group is because those two groups act as the coupling mechanism. Remember the other day I told you about the train with the thing that holds the train cars together? Same thing here. Here's amino acid number 1. Here's amino acid number 2. We're attaching amino acid number 3. Guess how it's done? Dehydration synthesis. Right? Water's created. So now we got 3 monomers, the 2 amino acids that were connected before, the third one, and now we've got a polymer of 3 monomers, in this case, amino acids. See what I mean by the purple part? The carboxylic acid and the amine groups are necessary because what you're wanting to do with all these amino acids is link them together. You see, we're creating a big puzzle piece here. And it works the same way. It's dehydration synthesis. Now, the peptide bond. Peptide is a term specific for proteins. You might sometimes actually see proteins called polypeptides because that's what they are. They're a bunch of peptide bonds. A peptide bond chemically is a covalent bond. It's a solid line, just like you're used to. The specificity, there we go. The specificity of it being called a peptide bond is it's between 2 amino acids. All right? So a peptide bond is a covalent bond specifically connecting 2 amino acids. And now you're getting the backbone of the protein and all those side chains. And remember, the ones that were yellow were hydrophobic and the green ones are hydrophilic. So when you put them in the water, you know, this one here is going to want to bend and get away from the water, and these are going to be attracted towards the water. Now you can imagine a chain of, oh my God, protein's going to be thousands of amino acids long. And you can imagine all these chains interacting with water is going to get that long chain to start folding and kinking up and forming all these really cool shapes that to us look like blobs. There's the ribbon model, so that's a chain of amino acids. And notice how they're making all these kinks and curls and you know, twists and turns. And then the space-filling model, and then what we call a wireframe model. But notice the important part is a shape. In this particular picture, oh my God, this is so cool. The way this thing folds up, it creates a groove right here. I guess I should probably use a white pen. There we go. See that groove? Now that groove has a particular shape. Doesn't look like a shape to you and me, but if you were the target molecule, you would fit very nicely in that groove. Now, why is it fitting in the groove? Well, don't worry about that right now. It's fitting in a groove for reasons that we'll talk about when we get into enzymes. But you might be able to think about one reason that proteins would have a shape. You remember when I was talking back about your defensive proteins, like antibodies? How does an antibody recognize, say, for example, the flu virus? How does an antibody protein, which is going to get rid of the flu virus, how does it know of all the things floating around in your body? How does that antibody for the flu virus know that I've come across a flu virus, I'm going to destroy it? How does it, quote unquote, recognize it? I'm going to ask you a similar question about something you're more familiar with. How come the key to your car will only open your car and not mine? They're both keys. What's the most important thing about a protein? See? Does that make sense? There's the protein from the flu virus. Got a shape. It looks kind of snake-ish, squiggly. But the antibody protein, look at its shape. Perfect complementary shape to that, isn't it? So when these things are bumping around randomly, when they get close enough, the reason why they're going to recognize each other, even when we talk about enzymes in subsequent chapters, the way these proteins recognize the thing that they're supposed to interact with, in this case, the flu virus, is shape. And the only reason that you get that squiggly, weird shape where you do is because you put the right amino acids in the right order. Because remember, again, we live in an aqueous world. So what's going to happen is that protein, once it's created, is going to be dumped into water. And because of the hydrophobic and the hydrophilic amino acid, it's going to go, blah, 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 blah. If you could see the camera, see my hands go, blah, 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 it's going to fold itself up. And it's going to make a particular shape. Now, if it doesn't get the right shape, you're not going to get rid of the flu virus. That's how antibodies recognize the disease. That's when they give you a vaccine. That's what they're doing. They're giving you a little bit, in some cases, they're giving you a little bit of the virus so that your body will produce those antibodies. Not enough of the virus to really make you sick. Although sometimes that's why I never get flu shots. They always make me sick. I don't always get the flu every year, but I'm guaranteed to get the flu at least once if I get the flu shot. But that's what it's doing. Your body's building antibodies. So if you come across it again, it recognizes it. And it doesn't go, oh, old friend flu virus. It recognizes it because of shape. All right. Have I pounded the shape thing enough? Are you guys done hearing about shape? Or actually the importance of shape? Because now I want to show you how you get this very important particular shape. And this is such an incredible story. I want to make sure everybody's on page here, right? So it's shape. Let's talk about how this happens. All right, take a look at this picture here. This is a protein. Fever is probably one thing. Flushing out your digestive system might be another reason why they tell you why they want you to do that. Yeah, and of course you want to remain hydrated, and the fever might cause that. And if you have diarrhea or vomiting, then you're becoming dehydrated. That's how folks die of cholera. Cholera is a disease that kills you because you basically dehydrate by having excessive diarrhea. It just seems like a wonderful way to die, huh? Okay, you guys taking in this picture here? This is a protein. It's got a particular name, transthyretin. You can look it up, figure out what it is. I have no idea, but it is a protein. I think I looked it up once. I just don't remember. Is this the one? No, that's the other one. There's a protein in your tears that actually helps your body protect from foreign things getting in through your eyes. It's a defensive protein. Okay, proteins have 4 levels of structure, all right? 4 levels of complexity to their structure is what I really should say. And the first level is the primary structure. And what we mean by the primary structure protein is, you can very clearly see it's illustrated here. The number of amino acids, this one's got, 127, and which amino acids, right? So you can, and again, don't worry about the abbreviations. I'm not going to ask you to memorize amino acids, but number 1 is GLY, that's glycine, proline, threonine, then another glycine, and then another threonine, and so on and so on and so on and so on. Okay? Now, it's kind of a little bit of a foreshadowing towards the end of the semester. This is really cool. This recipe for this particular protein, transthyretin, this 127 amino acid recipe, that's your genetic code. Your body knows which amino acids to put in each of those spots, spot 42, spot 71, because your genetic code is telling you to. Isn't that cool? That's the recipe. So what you're looking at here, and that's the neat thing, you can actually figure out a DNA of an organism by looking at the primary sequence of proteins. If you know the amino acids that are there, you can actually trace that back and figure out what the DNA code is. And I'll show you how to do that later on in the semester, but that's some neat detective work. Okay, so the primary structure of protein is the sequence of amino acids. Make sense? Real simple, right? Now, if this gets screwed up, if you get the wrong amino acid in the wrong order, right, you can potentially mess up the protein. I said potentially, not necessarily, but potentially. Okay, like for example, if you switched one amino acid for another, but they were both hydrophilic, the effects may or may not be extreme. If you switched a hydrophilic amino acid through some problem, instead of a hydrophilic one, a hydrophobic one was put in that place, problems might be a little bit bigger for reasons that you should be able to understand, right? The hydrophilic part is going to cause the protein to fold towards the water, but you've stuck in a hydrophobic part, so now that's going to fold away from the water. And that's going to cause a different shape, right? Okay, primary structure. Now the secondary level of protein structure. And I want you to understand that each of these levels is built upon the other one. The secondary protein structure involves two, let's call them varieties. All those little stick models you see there, folks, don't get confused. Those are all just amino acids. Because of the amino acids being where they are, because of that primary level of structure, if that is proper, then throughout that huge chain of amino acids, you're going to get some areas, because of our buddy hydrogen bonds, because of what side groups are there, you're going to get Sorry, folks. Y'all still with me? I just went out of the classroom for a second. Hello? Oh, you there? What the heck was that? Anyway, so because you have the right amino acids in the right order, what's going to happen is throughout that protein, you're going to form this spiral shaped thing, this alpha helix. A helix, a spring is a helix. Anything that twists like that. Curly Q, what kind of pasta is that? Is that rotini pasta? That's a helix, okay? So because you have them in the right place. Now, again, you're not going to get those helices, that's the plural of helix, in the right place if you don't have the right amino acids. Remember, it's all built up. If the primary structure is messed up, it potentially is going to mess up everything else. The other variant, so this is one here, is something's known as a beta. This is a beta pleated sheet. So what's going to happen in a beta pleated sheet is because the amino acids are being where they are, because of those wonderful hydrogen bonds, side-by-side chains of amino acids are going to form this a pleated sheet, a pleat, like if you folded something back and forth. You can see in the picture they're folded, right? Now, you might be thinking, again, alpha helices and pleated sheets, but remember, these are shapes. And to Mother Nature, those shapes are real important. Having those alpha helices and those beta pleated sheets, those beta strands in the right place is really crucial. Let me show you an example of a protein. I'm going to show you a picture of a protein, the ribbon model, like we saw in that picture a few pictures ago. This is spider silk. I love spiders. I think they're cool. My son's not crazy about them. This is spider silk. Spider silk is a protein, actually a pretty strong protein, right? Whenever we go bike riding in the woods, he always makes me go first, so I go through all the spider webs because he's arachnophobic. I tell him he's a web, but he makes twice the money I do, so I guess he's not that much, right? Now look at this protein. It's huge. It's long, but notice a couple things. Let me get white. Easier. There's an alpha helix there. A lot of alpha helices. There's one. I think my pen just died. Anyway, there's an alpha helix there. Any of the purple, right? In the green, these are the pleated sheets. guess that's supposed to be green, didn't come out too green, but the green are the flat areas. Those are those pleated sheets, right? And again, if you don't have the right amino acids in the right order, you're not going to get those helices. You're not going to get those pleated sheets. You may not be able, if I'm looking at this protein, you may not be able to get the right shape to actually create that spider web. And like I said, if you know, proteins are pretty strong, and spider web, in spite of the fact that it's real thin, it's pretty strong stuff, right? Very good structural material. All right, now, if you'll notice in this protein, because we're going on to the next level now, it's not just a matter of having these curly cues and these fully pleated sheets in the right place too. You notice this protein, you could stretch it out in one big long chain, has folded back on itself. It's folded, it's twisted, it's turned, right? Now that twisting and turning and folding back, again, is going to be because of the side groups. If you take a look at the tertiary level, these are just some examples. So again, we got the ribbon model here, and what we're looking at, what we're looking at, I got to try to find one of those quadruple A batteries for my little pen, which apparently only exist on Amazon. So this ribbon right here is the sequence of amino acids. So these are the side group, or no, I'm sorry, these are the functional groups, and these are all side groups. So there's one here, and then we move down. Let's skip this one for just a second right there, and we're coming down here. There's another amino acid there, so there's one there, and it has this side group. The same with these guys, and they have those side groups and here. Anyway, you get my point. Because those amino acids that I'm marking with X's have the side groups where they do, what they're able to do is engage in chemical reactions. The tertiary level, the third level of protein structure, right, we're building upon them, the tertiary level, is caused by the chemical interactions of the side groups. All right? For example, the hydrogen bonds, the disulfide bridges, the ionic bonds. Okay? Those things would actually pull that ribbon together. See, the hydrogen bond, that's attractive force. The disulfide bridges, remember I told you the other day that if you have a lot of sulfhydryl functional groups in the protein that makes up your hair, you have kinky hair. So that's what it causes. It causes disulfide bridges in the protein. That's why you get that kinkiness. That's not straight. It's folding it back and forth. Ionic bonds also attract things. Now, look at the ones up here, though. Instead of being attracted to each other, these two things are actually going to come together because of a common enemy, maybe. They're hydrophobic. So if you take a protein, like in the picture up here, and you put it into water, all of the hydrophobic parts are going to be on the inside, trying to get away from the water, and all the hydrophilic parts, because of the amino acid side chains, are going to be facing outside. Absolutely brilliant, isn't it? You put the right amino. I love the random, not the randomness, but just the idea. You lick a bunch of amino acids, you throw it in the water, and bop, bop, bop. It's kind of like Transformers, right? Remember those things that maybe you played with or your kids played with? You can make them into a boat. You can make it into a plane. Oh, I love this. This is so cool. All right. Any questions on that? So you can see why I'm saying that if you get the amino acids wrong, right? Yeah, if you get the amino acids wrong, it all gets truncated forward and the shape gets messed up. Not necessarily in a bad way, but you know, when you're doing things like that, you never know. All right. Now, many proteins, this is as far as the complexity goes. But there are some super proteins out there. There are some proteins like collagen. Where do you find collagen in your body? If I asked you to grab a body part with collagen, what body part would you grab? I could show you if I had a video. Grab your lip. Grab your lip and do back and forth. Your lips are spongy, aren't they? They're elastic, huh? Your lips have this rubbery, well, maybe not rubbery, but this sort of, rubbery-like texture. They're springy. Proteins are that way. Proteins are very springy. They're very good at holding things together because they give a little bit. They're like a rubber band, so they're not real hard structures necessarily. Collagen is actually a super protein. It's made up of 3 subproteins, the red, the green, and the blue you see in the picture. Collagen is 3 proteins, each with a primary, secondary, and tertiary level, and then they bind together. This is what we call the quaternary level. Not all proteins have this, just those super proteins that are made up of subunits. Like the one down below, that's hemoglobin, the protein in red blood cells. Hemoglobin is responsible for transporting those, carbon dioxide and oxygen throughout your body. And it's actually made up of 2 alpha unit proteins and 2 beta subunit proteins. So it's actually made up of 4 proteins. See the iron molecule there? That's why iron is important for blood. Iron helps hold the heme part together. The heme part is the part that actually attaches to the oxygen in your lungs and the carbon dioxide. It's actually what does the transporting part. Okay? All right, now let me show you an example here of how important the structure of a protein is. You guys are probably all familiar with the disease sickle cell anemia. Sickle cell anemia causes anemia, which is low oxygen, right? And physically, it causes many of the red blood cells in someone with sickle cell anemia will have this sickle shape to them. The problem with sickle-shaped red blood cells, anemia is a problem, of course, the lack of oxygen. Yeah, yeah, there you go. The problem with them too is they don't flow very well through blood vessels. See, your normal blood cells have a nice, see the normal ones, they have a nice round shape, and they're actually kind of round and spongy. So they can actually fold and roll around each other, and they move along very smoothly. But the sickle-shaped ones don't. Now, the difference between having hemoglobin that's normal and hemoglobin that ends up giving you sickle cell anemia is, if you look in the primary structure at amino acid number 6, if you have normal hemoglobin, that amino acid is glutamic acid number 6. If it's sickle celled, it's valine. Now you're thinking, one amino acid wrong? And there's hundreds of amino acids in hemoglobin. All right, folks, glutamic acid is hydrophilic and valine is hydrophobic. So what happens when you have glutamic acid is you get normal hemoglobin, and the proteins, the alpha and beta proteins, they don't associate with each other. So each one carries oxygen and everything flows along nice and smoothly. When you have the valine in there, that causes those beta proteins to be misshapen. You see that? Get that sickle shape? Causes those proteins to stick together. And actually, become fibrous. And what that does then is that not only that's the anemia part, that only slows down their ability to transport oxygen, but as again a physical thing, anemic cells don't flow very well, and folks with anemia oftentimes suffer lower blood flow to extremities and things like that. Now there is a thing called sickle cell anemia trait. Yeah. Do you know why that is? Do you know why we predominantly find sickle cell anemia in African Americans? It's a cool little detective story. You do? What is it? Okay. Sickle cell anemia, of course, is a genetic trait. It's part of your amino acids, which means it's part of your DNA. And African Americans, like typically what happens among folks of an ethnicity, yeah, you probably did talk about it, tend to breed with folks of their, especially in the old days. You know, if you lived in Africa, you didn't travel to the other side of the world for a mate. You pretty much mated with someone nearby. So ethnic groups and social groups tended to breed amongst themselves. So certain genetic traits in a particular ethnic group would actually be enhanced if you continue to breed. Now, I'm not talking about inbreeding. I'm talking about a group of folks that aren't related to one another, but that's the breeding stock, if you will, the breeding population. The reason why sickle cell anemia, so that's why it's higher in African Americans today. But historically, the reason why that is back in Africa when human beings were dealing with the disease malaria, folks with sickle cell anemia, more specifically, folks with sickle cell anemia trait. If you have sickle cell anemia trait, some of your red blood cells are sickle shaped. Fewer of them are if you have full-blown sickle cell anemia. Folks with sickle cell anemia trait have to be careful in low oxygen places. Ryan Clark used to play for the Pittsburgh Steelers. I'm not sure if he realized it, but he had sickle cell anemia trait. And when they went and played in Denver, and remember Denver is a mile high, he suffered. He had to end up going to the hospital. He had problems because of his sickle cell anemia trait. But anyway, back to the reason why sickle cell anemia trait is, or having sickle cell anemia trait or sickle cell makes you immune to malaria. It's actually a protozoan. It's a protozoan parasite that gets into you, and it causes you to, have malaria. And sickle-shaped red blood cells are more difficult so they actually have increased survivability with malaria. All right, I got a couple of more slides. We'll finish up proteins and we'll do nucleic acid. So we will move the exam to Wednesday. I will send a notice out to everybody. Oh man, where's my notebook? Write myself a note right now. I got to remember to do the India ink, and I also got to do the exam. So let me write India ink. Oh, my wife didn't need this. All right, now. Protein shape is very important. Protein shapes can be changed. Typically, heat is one of the more common ones, but acids and bases can do it too. pH can do it. When a normal protein has been exposed to excessive heat or to an area that's too acidic or too basic, it will denature. The heat or the acid and the base will cause those side groups, that tertiary structure and that quaternary structure, start falling apart. Now, a denatured protein doesn't have the right shape and it's useless. But that's not always the end of the story. Sometimes proteins can be permanently denatured. That's why I have the picture of the egg down there. When you heat that egg white in a skillet, it becomes permanently denatured. If you let that egg cool off, it wouldn't go back to the translucent soupiness of the egg white before it was cooked, right? But some proteins, because they're springy and because they're very important and expensive to make, folks, your body uses a lot of energy and materials to create proteins. They are resilient enough that if the heat, if it cools off, they can renature themselves. Now, we'll discuss this a little bit more when we get into enzymes, because enzymes have a particular shape and they need to have that shape to bind to the molecules they work on. So if you denature an enzyme, you affect its ability to do its job, right? Okay, I'm over time, so I do have one more protein slide, but I'm not going to do it today. I'll pick that up on Monday. So again, folks, don't forget the exam, chapters 1 through 5 will be on Wednesday morning, not Monday. And additionally, recall that your assignments, your scientific method assignment is due. We'll make it due on Wednesday at 8 instead of Monday at 8. Okay? All right, anybody have any questions for me? Let me stop the recording.